Thanks very much, Claudio. It's really great to be here, and thanks for the invitation. Let me see, can I get this to work? Can't share screen. Okay, let me see. A window, maybe a window might work. Okay, you know what? I have a problem, Claudio. I'm on Firefox and it's not allowing me share my screen for some reason.
Huh. Could you switch to Chrome?
I'll quickly go onto Chrome if that's okay. I'll be back in like 2 seconds. Okay.
Okay.
Actually, apologies. I didn't know there was going to be. I just resent the world over by Google, and
all right.
I'm not a Chrome person, but this happened once before, and it went fine when I went onto Chrome. So let me just take a second. Okay. Okay, can you hear me, Claudio? Yeah.
Yes, we can hear you.
Can you see me? I can't see. Oh yeah.
We can see you, not the screen yet.
Okay, let me see. It's coming now.
Okay, good.
Can you see that okay?
Yes, it's fine.
Okay. There's a lot of feedback. I don't know. Hi, good morning, everyone. It's a real pleasure to maybe to be here. Let me see what the joys of online technology. I'm just going to stop sharing for a second and then share again because I can't present now my entire screen. There we go. That's better. Okay, so it's a real pleasure to be here, and I'm going to take you through the journey that we've been on in Cork, in the very south of Ireland, for well over a decade now. And in this presenting mode, I can't see anyone else, or I don't even know. I hope everyone can hear me okay. I'm sure you'll jump in if you can't. So I work in a medical school, and in medicine, everything more or less goes back to Hippocrates, and Hippocrates is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. And this can be quite surprising, you know, when I try and convince my psychiatry and neurology colleagues that there might be that this might be the case. And also, you know, when I talk to my neuroscience friends, they wonder why, if I'm so interested in the neurobiology of emotions, which I am, why am I so fixated about what's going on in the gut? But all we have to do is look to our language. We use phrases like gut feelings, gut instincts. We make gutsy moves when we're brave. We're gutted when we're disappointed. We have gut instincts. We have butterflies in our tummy when we're nervous. So we personify all of our emotions within our gut. And what we've been interested in to see is what is the underlying biology of this. Now, let me introduce you to Eli Metchnikoff. Many of you may not know who he is, but he's got one of these kind of heroes in this story. Metchnikoff was, if you're an immunologist, of course, you will know who he is, because he is one of the founders of modern immunology, having won the Nobel Prize for his seminal work on discovering the process of phagocytosis, how immune cells gobble up foreign bodies. And as often happens, great scientists, later on in their careers, they can start coming up with some crazy ideas. And Metchnikoff was full of these ideas. And one of these ideas was, why did people in what's now parts of rural Bulgaria, why did they live longer and healthier? And so he started exploring this and carrying out some naturalistic studies to really understand it. And what he put it down to, it was what they ate. And what they ate was a lot of foods containing lactic acid bacteria, a lot of fermented foods. And so not only is he one of the fathers of immunology, he's one of the fathers of geriatric science in terms of systematically recording how healthy aging can occur. And he is also one of the fathers of probiotics. And so if you go to Korea, you can get a yogurt drink with his picture on it. So it must have that Nobel sign of approval, and it must be good for you overall. Anyway, most of what Metchnikoff was doing was forgotten about for about 70 or 80 years. But more recently, people have got really excited about some of these ideas in the context of the microbiome. And you'll have just heard from Antonio all about the role of the microbiome in many aspects of health. But I'll just reinforce some of the key messages, because repetition is really important here. And there are a few things that I want to remind you. One is that we are living in a microbial world. Microbes were here long before humans, and therefore it's important to have everything in that context, from an evolutionary context. If we look at genes, we are more than 99% microbial. All of the money we spent on the Human Genome Project, and it's for less than 1% of our genes. It's quite humbling. It's also quite humbling as a neuroscientist to think that the weight of our microbes is roughly the same as that of our brain. Now, if I'd been given this talk a number of years ago, I would have told you, and you still see it written in the literature, that we have 10 times more microbial cells than we do human cells. But that has been revised downwards. And so it's now thought to be about 1.3 to 1. So we're still more microbial, but just about. So just remember that the next time you go to the bathroom and you get rid of some of your microbes, that you're becoming more human, and what that might mean. When we talk about the microbiome, for the most part, we don't mean the microbiome at all. We actually mean the bacteriome, because that's what we're measuring. But we can't forget the other aspects of this really rich ecosystem we have in and on our bodies. And these include the viruses and bacteriophage, the archaea, the fungi, protists, etc. So a lot of this work is still bubbling up and is lagging behind the bacteriome. It is one of the hottest areas in biomedicine, making the cover of all the glamour magazines, and the lay press just love it in different ways. And what we're interested in our lab is really trying to understand the role of this microbiome across the lifespan in modulating brain function. And we know that the brain changes from early development in utero all the way through early life, and then you start to see different changes again occurring in aging. And so you can parallel some of these same changes that are occurring in the microbiome during these key periods, and one of the goals, and I won't have that much time today to go into it, but one of the goals of our lab is really trying to understand at a mechanistic level is what are the signals coming from the microbiome at these key windows, and how they're contributing to shaping and sculpting and neural circuits that might be important for a variety of brain disorders. For the most part, it's thought that we're sterile in utero, and we get our microbes kind of as a birthday present from our mothers as we emerge through the birth canal. And therefore, in the first 2 years of life, there's many factors that can influence the composition of this microbiome. Among them, of course, is mode of nutritional provision being one of the major ones. The host genetics, the environment we live in, exposure to antibiotics. One of the areas we've been interested in a lot in is mode of delivery at birth. Because if you're born by C-section, then you don't get this handover of an evolutionary baton in a relay race of life from your mother directly. You get it more from the hospital environment or the skin. And this goes back to early work from Gloria Domingos-Bello and her team, which showed in this pilot study that there was a separation between infants born by C-section and those born vaginally. This has been reproduced now in at least 14 large cohort studies, most notably this TEDdy study, where there's 12,000 individuals. And they showed two major factors influencing the composition of the microbiome in early life. One is breastfeeding. The receipt of breast milk was really shaping this microbiome. And secondly, birth mode. And so that's really important. And so we're beginning to see that the microbiome in early life is really an important security blanket in shaping and programming the immune system and all of the other systems within the body. Moreover, work from Trevor Lawley's group more recently has shown that in C-section, we also, not only do we lack some of the microbes that we get from our mothers, we also get a blooming of some pathogenic bacteria. Now, we know that being born by C-section increases your relative risk of asthma and allergy and type 1 diabetes. We know very little about it in the context of brain health, and this is something we've been studying in conjunction with our epidemiology colleagues and doing some human studies, but also more recently doing some animal studies, where this is work from Livia Moret, who's now in Sarkis Mesmanian's lab in Caltech, but as for her PhD, she was working on developing an animal model of C-section, where animals are either born vaginally or compared with those born by C-section. And when these animals grow up, they have alterations in key behaviors relevant to stress, anxiety, and social behavior. They also have really marked changes in their microbiome early in life, and particularly in relation to bifidobacteria. There's a reduction in the numbers of bifidobacteria. So one of the strategies we have in this model is we've been looking at ways, how can we rescue this phenotype? Now, mice, like other mammals, some other mammals are coprophagic, so they eat each other's poo. So one of the ways we did was we were able to do kind of a artificial FMT by co-housing the animals together, and that was able to rescue the phenotype. Now, that's not applicable from a human perspective. So then we've been looking at other strategies, either through giving specific strains of bacteria or specific prebiotics. And really what we're thinking about from a nutritional point of view is, because C-section is an important life-saving intervention that happens in at least 15% of the population by medical mandates. It's happening at much higher rates in many parts of the world, including in Ireland and in Italy, but most notably, really in parts of Brazil, it's up to 70% elective C-section. In parts of China, it's at 60%. So, but in the when C-section is mandated, what can we do, and can we offer other interventions? And our animal studies, at least, are offering hope that perhaps you can, through nutritional interventions targeting bifidobacteria, we can reverse some of the effects of C-section. A paper came out last night from Willem de Vos's group showing that FMT is another approach that could be used for C-section induced effects. So this is quite exciting, and we're doing a lot in this area. However, my neuroscience colleagues are quite a skeptical bunch, and so they need more evidence that the microbiome is important in early life. So how do you get more evidence that something is important? Well, looking to engineering or other aspects of biology, one of the best ways to get evidence is to take it out and see what happens. And so therefore, the concept of growing up germ-free without bacteria, well, that goes back to Pasteur's times when they were trying to develop this. Here's a germ-free man in a sci-fi magazine from the 1920s. Now, he doesn't look that great. Our germ-free mice, while they have their own problems, they look a lot better. But one of the things they do not do is mimic any form of human, really, translation. They grow up in a bubble, and perhaps, you know, with the exception of the boy in the bubble that Paul Simon sang about in the 1980s. But they have allowed us to be able to answer the question, is the microbiome involved, yes or no? And they've taken us to some really weird and wonderful places in neurobiology that we didn't expect to go. For example, we showed that when you knocked out microbes during development, what happens in the prefrontal cortex is a very specific increase in myelination ongoing. Now, myelin is the insulation on which nerve cells have for appropriate conductance. You may hear about it in the context of demyelinating disorders like multiple sclerosis. But we found that basically the microbiome was shaping prefrontal myelination patterns. By looking down the electron microscope, we could really see this. And so this offers the opportunity that potentially through nutritional interventions that we could actually target the microbiome to support brain health at key windows. Moreover, adult hippocampal neurogenesis, the birth of new neurons. This is a really important process that happens in the hippocampus area of the brain, an area of the brain involved in learning and memory. And this happens throughout our lifespan, and it starts to diminish as we get older. And these new cells that are formed are really important for complex cognitive functions like pattern separation. They also have been implicated in the stress response and in antidepressant action. And so we've shown that in the absence of microbiome, the regulation, how these cells are working is completely gone awry. We got changes in a variety of neurotransmitters and in synaptic plasticity, and we're now doing really important studies at the electrophysiological level to back this up, as well as changes in dendritic growth and arborization. Work from Marco Prince's group and Sonia Gorel's group and others have shown that microglia, the brain's immune cells, macrophages, are not functioning appropriately. And as well, work from Sven Pettersson's group is showing that in germ-free animals, there's a deficit in the blood-brain barrier. So here, you don't need to be a neuroscientist to be able to see here very clearly. This is a neuron, a nerve cell from the amygdala, a region of the brain involved in fear, in anxiety, in the emotional aspects of pain, and in the social response. Now, the amygdala, so when we look at the amygdala neuron here in germ-free animals, it's much more arborized. It's almost like in a chronic stress state already. And what does that translate to in behavior? And I won't go into the details, but it translates to changes in these amygdala-dependent behaviors. We get deficits in amygdala-dependent fear learning. The animals are not able to recall appropriately. Changes in abdominal pain. Abdominal pain is a core symptom of irritable bowel syndrome, one of the most unloved disorders we have out there, IBS, and most poorly understood, and one that is so common and offers so much distress to patients. And we find again that visceral pain has a strong link with the microbiome overall. And then finally, we were interested in social behavior. And mice, like humans, are quite social. So if you give them the chance to spend time with an empty chamber versus a mouse, they'll gravitate towards the mouse, but not if they're germ-free. Mice, like perhaps some humans, are a bit fickle. So if you give them the chance to spend time with a new playmate versus their everyday playmate, they'll normally prefer the new playmate. Again, not if they're germ-free. And so we're beginning to see that for normal social behavior, and if you're a mouse, of course, that you need to have appropriate microbes in your gut. Now, what we've started then to look at, and this has been a really nice work, was to try and look across the animal kingdom when we start to affect the social hierarchies, social structures, social behaviors by either artificial or natural means. What we find is that we start to interfere with the composition of the microbiome. And this goes all the way from insects all the way up to non-human primates. Just think over the last 6 months, what's happened in Italy and around the world in terms of how we've interfered with social behavior through lockdowns, through various restrictions and social distancing. And the question remains is, what is the long-term impact that has on our microbiome, and how our microbiome is signaling to the brain? Because also we're finding there's something about the social brain that makes it very appropriate for microbiome interventions and microbiome signaling. And this may have a lot of impact to be discovered yet in a variety of disorders of sociability, whether it's in autism or in the social aspects of schizophrenia, or anxiety disorders of sociability, like social anxiety disorder, which we're quite interested in studying at the moment in the lab. So let me move to the adolescent period. So away from early life programming, but now to adolescence. And so I'm the professor of anatomy, and so I always like to highlight that and any of you with teenagers will know this already, that the anatomy of a teenage brain is very different, and it's really much a brain that's under construction. And this is one of the most exciting areas in all biological psychiatry right now, is trying to understand what is going on here during this critical window that is basically giving susceptibility to a variety of disorders that start to emerge here. We're, a lot of focus now on how microglia and neurons are interacting in the brain overall. Moreover, there's a lot of focus on various insults that start to occur to the adolescent brain, whether it's through stress, poor nutrition, alcohol, inadequate sleep, or drugs abuse. And a lot of focus is on what they're doing directly to the brain. But I would contend that all of these can also affect what's going on in the gut and in the gut microbiome, and then will change gut microbiome brain signaling. And so we have a lot of exciting work going on in this field, including some ongoing work now in the area of binge alcohol intake in humans to really start to tap into this overall. But going back to some of our early animal work, we showed some years ago now, 5 years ago, that if you deplete the microbiome during this critical window, you start to get deficits in anxiety, in cognition, in social learning. And more recently, we started to show that manipulating the microbiome through bad diets, in this case, we give high-fat diets or a westernized cafeteria-style diet. What we found is it really messed up the microbiome a lot and changed a lot of the genes in the amygdala that are involved in neuroinflammation. The behavioral effects weren't so strong, but we did get these priming effects. And so the question remained as whether there would be like a susceptibility being borne out by bad diet during adolescence that could be retained moreover. And again, we're getting changes in myelin in the prefrontal cortex, especially with the high-fat diet and these changes in the microbiome that are occurring. But there's a flip side to this, and this is a wonderful collaboration we had with Beatrice Pisani's group in Florence, where what if you could target adolescent period with a good diet, and could you then attenuate some of the effects of what's going on in the context of stress? And so in this paper, what we looked at was adolescent stress. And we find that there are deficits in learning and memory and using these two novel object recognition or contextual fear conditioning. So here in red is the stressed animals. And so when we fed them a diet that was rich in polyunsaturated fatty acids and vitamin A, we were able to attenuate these effects both in adolescence and in adulthood. So this offers a kind of a unique area of intervention through positive ways by diet and to actually mitigate some of the long-term effects of adolescent stress. And what we looked at was, what's this doing to the microbiome, and could the microbiome be playing a role? And it really looks like it is. And you can see here there are lots of changes that are going on in the microbiome in the diet that's given during adolescence. And so this is really been an interesting way that there is both pros and cons during this period of dietary manipulations. I mentioned alcohol. We've been working on alcohol and the impact that alcohol has had on the microbiome. This is a collaboration with Andrew Holmes's group at the NIH, and showing that basically what you start to see is a blooming of certain pro-inflammatory microorganisms in chronic alcohol intake. And we could also correlate this with changes in the brain, for example, in striatal dopamine receptor expression in animal models of alcoholism. And this is a collaboration with Ben Buttrell's group at the University of Lausanne. So let's skip to the other extreme of life, to aging. And, you know, with aging, and this is something, of course, all of my Italian colleagues are very aware of, the whole process of inflammaging. You know, inflammaging affects not just the immune system, but we have changes going on at the cardiovascular level, at the gut level, and at the brain level. And this is, of course, in line exactly what Metchnikoff was talking about all them years ago. And work from my colleagues here in Cork, from Paul O'Toole, some years ago now, showed that in an aged population that the microbiome was severely, you got severe reductions in the diversity of the microbiome in aging, and that these microbial diversity changes correlated with health outcomes, most notably frailty. And what was really important in this paper was they went one step further to see, well, what's driving the changes in the microbial diversity? And what they found was that it was changes in the diet. So those that lived in the community having a very diverse diet, full of fibers, that they had a more diverse microbiome and better health outcomes. And so there's a lot of focus around Europe right now on this type of health research in targeting the microbiome to support healthy aging. And this is really, really exciting and really important. We've been revisiting Metchnikoff's ideas in the context of brain health and trying to understand what are that's driving these behavioral changes, these social deficits, these memory deficits, these cognitive changes that we see in aging. And what we find is that in baseline, there's changes in intestinal permeability, which we can exacerbate by stressing the animals overall. There's also changes in inflammation, changes in at the microbiome level, and this is really, really cool. But it opens up a big question, because this is quite descriptive. Can we do anything about it? Could we target or at least in our animal models, could we target with a specific microbiota targeted diet to attenuate some of the effects of aging? So this is something we've been working on through a JPI project called Ambrosiac. And for this, I decided I'd focus on middle age, because for obvious reasons, it's a time window I'm very interested in right now, shall we say. And so with middle age, we know in the brain that we start to get a really accumulation of neuroinflammatory insults and neuroinflammation. And what we find is in white bars here, you can just see there's an increase in stress-induced immune priming, an increase in infiltration of inflammatory monocytes, an increase of microglia activation. Basically, the brain is starting to become vulnerable to the effects of inflammation and inflammaging that's going on. And so then we targeted this with giving a diet enriched with inulin. Now, inulin is a prebiotic. A prebiotic is something that's supporting the development of beneficial bacteria. And this inulin is very highly enriched in certain vegetables like leeks and artichokes and many other vegetables. And so when we fed these animals inulin, we were able to attenuate many of the neuroinflammatory aspects associated with middle age. So the take-home method, this is work that was done by Marcus Boehm, a postdoc in the lab, together with my colleagues Harriet Shellikins and Ted Dinan, who's my clinical colleague. So the take-home message from this is really that if you're thinking of having a midlife crisis, that maybe you should forget about the motorbikes and turn to the leeks instead. And this might be a better for your brain health, at least overall. So where does this leave us? Well, it leaves us with really trying to figure out the mechanisms. How is all of this happening? And this is something that we're really, really focusing an awful lot of effort in my lab right now, really trying to dig into the mechanisms. How are microbes in the lumen of the gut signaling to the brain? Well, we know that the enteric nervous system is really important. We know that various hormones and the immune system is really important. But I'll point out here the vagus nerve. Vagus nerve is a long wandering nerve that sends signals from the brain to all of the organs and then back from the gut back to the brain. So it's really an important pathway of communication. And we showed many years ago, together with John Bienenstock's group in Canada, that if you cut the vagus nerve, all of the effects of a specific bacteria were gone. And so this tells us, therefore, that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions in different ways. And this is at least one pathway of communication of what's going on. But there are others, and it's really important to understand that the microbiome is like a little factory producing all kinds of weird and wonderful chemicals that our body wouldn't be able to make otherwise. What it does is it takes substrates from the diet. These are like the raw materials coming into the factory, and then it breaks them down. For example, short-chain fatty acids, which give really important energy to colonocytes in the gut and to many other organs, come from the fermentation of a variety of dietary fibers. We've been interested in saying, well, can we just bypass the microbes and just go to the end products, a postbiotic approach? And we've done this with short-chain fatty acids and shown that they could reverse the long-term effects of chronic stress. And this is a really nice way of looking at this. Perhaps the best example of this, and one of the most surprising that people often forget about, is that human breast milk contains the highest complexity of sugars of all mammals. And the thing about these sugars, these human milk oligosaccharides, is that the infant has no capacity to break them down. They're broken down by the microbes. So our microbes have co-evolved with the host to be able to be the appropriate microbes to break down these sugars to produce chemicals like sialic acid, which are really important for brain development. So we're beginning to see this co-evolution between our microbes and our cells that is informing and really being an important aspect of where we're going. Now, I mentioned that stress is something that I've been working on a lot, and we've shown the stress in early life changes the microbiome. We've shown that in germ-free animals, there is changes in stress-related behaviors, which leads us to say, well, then could we modulate the microbiome to have beneficial effects on stress? And this brings us to the whole area of psychobiotics, a term we've coined here in Cork. And we've broadened it out now to be really targeting the microbiome for mental health benefits. And there are different approaches for doing this. I mentioned various strains of bacteria through probiotics, through prebiotics, like inulin, as I've mentioned, combining both, symbiotics. Postbiotics, I mentioned, like short-chain fatty acids, fermented foods, going back to Metchnikoff. That's what he was talking about. And paraprobiotics, even, do the bacteria need to be alive at all? Could they could the important factors be decorating their cell wall and then interact with the immune system? And so this is really an important area that we feel that we're at the beginning of a new revolution in terms of how we are able to get to understand mental health. But we have a long way to go. I don't know what the situation is like in Italy, but I know in medical training here in Ireland, our medical students get very little nutrition studies, and our dentists get more. And I think we need to, it's slowly changing. We need to really understand, both from a preventative medicine point of view, but also from a symptom management aspect, how important diet is for our overall mental health and our overall well-being. I think that's really important aspect. Do we have any evidence? Well, in our animal studies, we do. In all of these areas, we've shown that a variety of strategies have been able to reduce anxiety, affect the stress response, and modulate neurotransmitters in the brain, including GABA and serotonin. And that's been quite exciting. Over a decade now, we've been showing that. But most of what we try doesn't do anything. And that's an important area. It's important to show that there's huge publication bias in this field, and for most of what's available to clinicians and dietitians and various people have never been tested at all. So we need more evidence, and we need to create more evidence, and there's a huge need in the field. At the human side, we were very buoyed by this very small study we did a number of years ago, where we took a biflongum from our animal studies, and we showed that when we went to a healthy volunteer population and we brought them into the lab, and we have really nice ways of stressing people in the lab, and we look at their brain EEG, we could get a really good signal that this biflongum was actually having an effect. Work from Paul Inck's group in Tübingen was able to take the same strain of bacteria based on our advice, and they were able to show that when a person was in the brain scanner, was playing a virtual reality game, basically a ball game with two others, and all of a sudden the two other people start to ignore the person in the scanner. Now, our brain doesn't like to be ignored. It takes us back to all these really bad childhood memories. And so we get a stress response mounted. And the people that had taken the biflongum didn't have any stress response. Now, buoyed by these exciting findings, we took the most exciting strain from our preclinical platforms and tested it in humans as well. And to be honest, you can't get more negative data in a paper if you tried. It really did absolutely nothing. And so that leads us with this challenge in this field, but it's not specific to the probiotic field, which is, how do we translate these animal studies to human studies? And there's a long way to go to understand this. Here, I just threw in this. This has just came out in the last week. It's a very, again, it's an exploratory study, but it is a randomized placebo-controlled study where we looked at a certain prebiotic fiber, in this case of polydextrose, and we're able to show on a variety of cognitive tests, again in healthy volunteers, we're able to show a very nice pro-cognitive effect of a dietary intervention in these individuals, and we're trying to link this in with specific changes at the microbiome level. And what people are also doing is going out and doing cross-cultural studies, looking at, how the microbiome is influenced by diet in relation to our ancestors. So, for example, in Tanzania, people have looked at the richness of the microbial diversity that's there and correlated with the diet, which is very high in fiber. We've been able to chart, well, what has the introduction of agricultural practices done to our microbiomes through our diets? And we start to see there's a diminution of microbial diversity. And then in our Western world, not just in the US or Ireland, but also in Italy, we're beginning to see that through the introduction of processed food, through stress and antibiotics in the food chain, we're beginning to see an extinction of the microbiome. And the question remains, how do we get it back? Is there a way? to really get back these, and if not, then you can take all the fiber in the world, and if you don't have the microbes that are there to actually harness benefit from it, then we might have a problem. One of the ways we've been interested in this is, again, with Metchnikoff in mind, we've been looking at milk kefir, and this is working in collaboration with my colleague Paul Cotter at Teagasc, our National Food Research Centre here. And I won't go into the details, but we've been working on different kefirs, milk kefirs, and seeing if they could have beneficial effects on a variety of behavioural. And we've published this year, showing that they do have anti-stress and pro-cognitive effects, and also have some effects on some of the social deficits overall. And really what the field is beginning to open up is breaking down barriers between nutrition, between microbiology, gastroenterology, and brain science. This is exemplified by this framework document from Marcus Reichel of the Default Mode Network Imaging, and Jeff Gordon, the father of the microbiome in some ways, where they really are highlighting that really to nurture the mind, we really need to feed and nurture the microbiome in different ways. And in tandem with this, we're seeing in psychiatry a whole new discipline emerge, where we're beginning to see that nutritional psychiatry is really important. And I was happy to contribute to these two framework documents, which are really saying how do we improve mental health by what we're eating. And this is a kind of a Minute Gus, which is a European-based project where we're able to try and create a new food pyramid that would be based on evidence, that would be able to support what we're doing in relation to brain health. And it's not just psychiatry. This was the cover of the Lancet Neurology in February, and really, you know, we're beginning to see that nutrition can play a role in preventative as well as management of a variety of neurodegenerative diseases, and that the implication of the microbiome in these is really important, and we're moving forward with this. And you stay tuned for a lot of excitement in this field as we move forward. In our own group right now, Kirsten Birding-Harold is a postdoc and nutritionist who is really, we're really excited about the preliminary data we have on this, where we're actually being able to show that by targeting the microbiome through a nutritional and educational intervention that is high in fermented foods and high in fiber, where we're able to be able to support aspects of brain health in people with relatively bad diets and who are susceptible to stress, mainly our student population. If you know, they're the ones who are quite captive for this. And the preliminary data for there is really exciting, and we hope to have more data emerge on this concept of a psychobiotic diet where we're able to triangulate diet, microbiome, and brain health as we move forward. What we're knowing is that our state of gut will therefore affect our state of mind. A few general things that will probably come up in questions. One of the hurdles we have is trying to understand what a normal microbiome is, because we can't, you can go to your doctor and find out what your normal cholesterol is and what your normal blood pressure is, but we don't know what a normal microbiome is. But with longitudinal measuring occurring in different ways, we're beginning to see people measuring their microbiome over time, and sequencing costs are decreasing. We're really going to see a huge excitement in the area of personalized nutrition and microbiome targeted interventions, and a huge amount of innovation and entrepreneurship occurring, most notably out of Israel, but out of other places as well. And this is really important. A lot of understanding about the interactions with the environment we live in, host genetics. We do need to really say that the buyer needs to be beware. There's a lot of nonsense in the media, a lot of problems with the way studies are interpreted. And so these are just one example of how the same study could be interpreted by different people. But the good news is that unlike our genomes, which all we can do is blame our parents or our grandparents, our microbiome is potentially modifiable. And understanding what the constraints on this is really going to be important overall. So here are some tips for a psychobiotic life. What we know based on evidence, we know that increasing the intake of certain prebiotics, omega-3 fatty acids, polyphenols, fermented foods has really good beneficial effects on increasing microbial diversity and supporting the development of good bacteria, whereas decreasing the intake of certain foods that we know, such as artificial sweeteners, emulsifiers, processed food, and too much red meat, you know, is also quite good. We know, as we heard from Antonio, that aerobic exercise is really good for our microbiome. We know there's a large data studies, including work we have ongoing now on circadian rhythms, and the microbiome and sleep disturbances play a big role here. at a population level, we need to try and avoid C-section as much as possible and support breastfeeding and minimize antibiotic use. And I finally caved into my wife and kids' calls to get a dog, because of course it's good for our microbiomes to have a pet. We need to be careful about overselling the microbiome. There's a lot of hype in this field. Jonathan Eisen gives the Overselling the Microbiome Award. We don't know where we are on this hype cycle yet. Only time will tell. Lots of really amazing work in rodents, but that hasn't stood up to translation in humans yet. And correlation does not equal causation. And this is an important aspect of the field. And microbiome field is probably the worst for this. And so, you know, I like this website called Spurious Correlations. You can correlate the divorce rate in the state of Maine with the per capita consumption of margarine. Now, I'm Irish and we eat butter, so you can see if someone kept coming home with butter, they could start some rows happening, and people start to fall out with each other, and that could lead to divorce eventually. And that's kind of the stories that we're being constructed in the microbiome field, especially in the early days. A lot of small, underpowered studies, and really not a really good appreciation of the importance of nutrition and diet and taking this. And of course, everyone thinks they can be a psychiatrist if they can just deploy a few scales, and that really undermines a lot of the phenotyping that we have. And we need a lot of rigorous trials across the lifespan, and we really need to dig into mechanisms overall. So this has been the work of a really amazing team of people that we have, including many Italians from all over Italy, and my clinical colleague, Ted Dinan, a big shout out to, who has recently retired. And I'm leaving you with Hippocrates, who is also thought to have said, "Let food be thy medicine." And that also could be fake news, but maybe I'll change it to, "Let food for your microbes be medicine for your mind." And I'll leave you with this cartoon from the Daily Mail. So thank you very much. And it's been a real pleasure talking to you today. And I'm happy to take questions if there's time.
Thank you very much, John, for this great presentation, really fascinating. So I will leave now open the discussion to the audience. If there are someone in the audience that would like to ask some question to John about the presentation. Yes, Natalia.
Hi, hello, Dr. Cryan. I am Natalia, a PhD student of University of Teramo. May I congratulate you on an amazing talk. I would like to ask you a personal opinion about the gut-brain axis. Here in University of Teramo, we are really interested on fermented foods, in particular of bacteria, microbes that are carried on these fermented foods, especially we work with Lactobacillus plantarum. And I will ask you about, what do you think about these fermented foods or these microbes that we can find in these fermented foods as a novel therapeutic target for gastrointestinal disorders, but also neurological or developmental disorders? Thank you.
Thank you, Natalia. I think, and this goes for a lot of the field, we just need more evidence. The amount of really good studies in the fermented food area in brain health are very limited. And and what we need to do is kind of create this, and that's why I'm very excited about fermented foods. And my colleague Paul Cotter, who I work very closely with, is like one of the experts we have here on food and fermented foods in general. And so it's a really good opportunity to really dig deep into what is specific about certain foods that could have beneficial. Well, you know, why are they having positive effects and others not in certain populations and certain individuals? It's difficult to do these human trials. Many of these foods have specific tastes that are very difficult to do in a placebo-controlled way. So that makes it tricky. I've struggled with this myself. How do I've been wanting to do a Kiefer study, and really it's very hard to think about how you can do it overall. But I think there's a huge scope here. So in our psychobiotic diet, we've added a lot of fermented foods into this without prescribing which ones they have to take, but we're trying to look at this as well. And so I think we just need to do a lot more studies, both in animals and humans, to gain confidence that this approach is going to benefit beyond a placebo level. And that's kind of where we need to go right now. There's been a nice systematic review from Felice Jacka's group in Deakin in Australia on fermented foods for mental health out just this year. So I'm quite excited about it, but I'd like to see more evidence than at just the population level.
Thank you.
Thank you very much. Some other question from the audience? Aldo, do you have any questions? Annalisa is.
Yeah, I have a question too. Professor Cryan, thank you very much for this very nice and also very interesting presentation. I only have a question. I mean, I have many questions, but the one is, you have shown some studies focused on specific bacterial strains. I would like to ask you, how did you select this strain? How did you choose this kind of strains? From some in vitro properties, or from other studies, or from other medical evidences?
Yeah, it's a wonderful question, and really it's one thing that the whole field is struggling with, because whatever strain you choose is always wrong for some reason. So initially in our earlier studies, we selected the strains largely because they had good information that they did something in vivo to the immune system, so that they had some immunomodulatory effects, because we were very interested in immune-brain interactions and gut-immune-brain interactions. More recently, what we've started to do is then screen in vitro our strains for activity at a variety of receptors that we're really interested in. So including a lot of G protein coupled receptors. And so we've shown and we've published on, for example, the ghrelin receptor. The ghrelin is a hunger hormone. And so we were able to screen in vitro supernatants from a variety of different bifidobacteria. And then we were able to pick the ones that had the most activity at the ghrelin receptor based on that. And that's the kind of approach now. We're looking at a wide variety of receptors and targets that we're interested in. And then we can look at that. We also looked in at their ability to have effects on, let's say, short-chain fatty acids or modulate tryptophan metabolism, or other pathways, neuroactive pathways that we're interested in. So it's gone from being a little bit ad hoc to be more targeted right now overall to see what we're doing.
Thank you.
Thank you. So, Aldo, I guess you have a question, right?
John, thank you very much for this very interesting presentation. I have just a question, more or less. I would like a comment from you about the possibility to move our research from gut populating strains, gut microbiota, to food populating microorganisms, because we put our attention on bifidobacteria, Akkermansia, and so on, but fermented foods and food in general are a source of very interesting microorganisms. And the probiotic concept perhaps should consider this type of barrier, and perhaps this is the time to remove that. What do you think about?
I think it's a great point, and I think we need as much diversity in terms of where we source our microbes from, and food is a wonderful matrix for many of these that has evolved over thousands of years to be able to get these bacteria where we want to, and of course we would include food among our psychobiotic type of approaches, for sure.
Okay, great. Thank you very much, John. I just have a last comment, if I can, because you showed some mechanism about how microbes release neurotransmitter and can act on the brain. But since also is my field, I would like to comment on how the epigenome of the host can be involved somehow.
No, this is really exciting area. And so there's a few things. One is some of these metabolites that microbes make, like butyrate are having direct effects on the host epigenome because they're working as HDAC inhibitors, histone deacetylase inhibitors. So there's direct effects. Other aspects like folate and various other things are coming from microbes. There's a lot of really exciting work on the host immune system and host gut, gastrointestinal system. Beautiful work from Teresa Allengott and David Artis and other people in the States working on epigenetic changes by the microbiome, and I think that's really cool. There's less, way less on the brain side of this, and I think it's really, really exciting area. One of the puzzles we had was does, from a butyrate perspective, would butyrate get into the brain in high enough concentrations from a microbial source to elicit these epigenetic effects? And the answer is still out on that. I worry that it may not be. We had a grant on this once and we got it funded, but then as we dug into it, we found that it was less and less likely that it was having a direct effect. But it can have an epigenetic effect on epithelial cells or immune cells that could be indirectly affecting what's going on in the gut. I think this is a really important area, and build it into a nutrition component. So you have a neutro epigenome aspects of the microbiome gut-brain axis.